Hi everyone. This is our next topic in the cardiovascular system. This is on heart. So as always, we have our learning objectives. There are 2 pages of learning objectives for heart. It's a little bit more complicated chapter. So I'm going to cover some anatomy, even though you have an anatomy component of this as well. I always think that reinforcement is good. And it also, in case someone is listening to this before they do the anatomy portion, then it's not quite so confusing. So you'll see that a lot throughout this semester where I cover the anatomy in the beginning of each of the lectures, just to make sure that everybody gets all that information. All right, so your heart is located in the thoracic cavity. It's between the lungs in a space. That space is called the mediastinum, and it sits within the pericardial cavity at the base of the mediastinum. So you can see that outlined area that they're showing you there. The superior surface of the heart is called the base, and it sits up at the top. So normally we think of the base as being the bottom. In the case of the heart, the base is actually up at the top. Okay, it's about the level of the third rib. The pointy part of the heart is called the apex, and it is at the bottom. And it lies between the fourth and fifth ribs and points towards the left arm. The apex is right about diaphragm level. The right side of the heart is tipped slightly forward, and the left side is tipped slightly backwards. So it is not sitting in there straight up and down. And that's the point that you want to make sure that you understand. It's sitting kind of tipped to the side, and it's tipped a little to the back and a little to the front. So it's not straight up and down like they often show in pictures. It is correctly oriented in this picture. Your heart, if you would think about your heart like a box with four squares, like a big box, and just make 4 divisions in it. Each of those empty divisions would be called a chamber. So each side is going to have an upper chamber. You have a right side of the heart and a left side of the heart. The upper chamber is the atrium, and the lower chamber is the ventricle. Now, a little bit about sides, because not everyone teaches this in 210. I do, but not everyone does, and it becomes really important in this course. So when we're talking about rights and lefts, the sides, it is from the patient's perspective. So when I'm looking at that heart, the blue side of the heart is the right side. So if you look over to the labels, it says right pulmonary arteries, right pulmonary veins. That is correct. The left side of the picture, when we're looking straight at it, because it's like we're facing our patient, is the red side. All right. So a lot of things in this course require rights and lefts. So why is that important? When you're chart noting, that has to go in. Absolutely. And when those chart notes go to the coder, there has to be a right and a left. If there's not a right and a left, she or he cannot code it, and then the individual cannot get reimbursed. So they're going to kick it back to you, and you're going to try to remember 2 weeks ago when you were treating Mr. Smith, if it was his right arm or his left arm. You need to make sure that you start doing rights and lefts. Now, you can just put a capital R or a capital L. I'm perfectly fine with that. In real chart noting, you actually have to write it out. But for in here, for the sake of time, I'm fine with rights and lefts. So this is my motto: right and left everything. If you don't need it, I'm not going to count off. If you do need it, I am. It's going to be half the value of the question. So practice it the way you want to write it. So when you're practicing this of the heart, let's just say you're practicing chambers, because that's what we're talking about right now, and you say atrium, right? So it's got a little line pointed to the right atrium. Don't just say atrium, say right atrium. If the little line is pointing to the right ventricle, don't just say to yourself, oh, that's the right ventricle. That's the ventricle. Say the right ventricle. Say it the way you're going to want to write it. And so right and left, right and left, right and left, everything. This is going to be imperative in vessels, in lab. So let's get this really good habit started now, so you won't have to belabor this when you get into your program. All right, so back to your chambers. Again, it's like a box. You have 2 upper chambers that are the atria. You have 2 lower chambers that are the ventricles. Each atria is the receiving chamber, so it's bringing blood in right? So the right-hand side is going to be bringing blood in from the body, and the left-hand side is going to be bringing blood in from the lungs, and it pushes it into the respective or lower ventricle. The ventricles are the primary pumping chambers that are going to send the body out to either the lungs or the rest of the body. So a little bit about circulation circuits. There are two distinct but linked circuits. So they are separate, but they are tied together at the same time. They are the pulmonary and the systemic circuits. The pulmonary circuit is going to transport blood to and from the lungs, hence the name pulmonary, where it picks up oxygen and delivers carbon dioxide for exhalation. The pulmonary circuit is a short pathway. It's not very far to go from your heart out to your lungs, and it is under low pressure. We don't want that blood to be under high pressure when it goes out to the lungs because it could do a lot of damage to the delicate lung tissue. So looking at our picture, we're going to look at the right side of the heart. That's blue, right? And we're going to go over this pathway here, but you can see that that blue side is going to go out to the lungs where it's going to pick up oxygen and turn red, and it's going to come back to the left-hand side of the heart where it's going to pass through and then go out to the tissues, drop off that oxygen, and then head back to the right-hand side of the heart. So blood is going to actually go through the heart 2 times. It's going to come in, go out to the lungs, come back, go out to the body, come into the heart, go to the lungs, come back, go out to the body. So it's always doing this process, and it's doing both sides at the same time. The systemic circuit is going to transport the oxygenated blood out to the rest of the body and then return the deoxygenated blood and the carbon dioxide to the heart, which is going to take it over to the lungs, which is going to get rid of that and reoxygenate it and bring it back in. The systemic circuit is a long, high pressure, high friction circuit because it's got to go all the way out to all of the parts of your body, including all the way down to your little toe, which is the furthest it possibly could have to go. From there, blood is going to be sent to the pulmonary circulation, as we've talked about. All right, let's talk about a little bit of the details now of this pathway. So blood moving out of the heart. So let's look at the right ventricle. So that is the blue side at the bottom. It's going to pump deoxygenated blood into the pulmonary trunk. The pulmonary trunk is going to split, or bifurcate is the big fancy vascular word for that, into the right and left pulmonary arteries, which are going to carry blood out to the lungs, and that is the first half of the pulmonary circuit, right? So going out to the lungs, and then it's going to come back to the heart. The left ventricle is going to pump oxygenated blood, so the left ventricle is going to be the red side down at the bottom. It's going to pass blood or pump blood into the aorta, and we're going to talk about the different parts of the aorta, and then it's going to branch and take blood out to the rest of the body. That's your systemic circuit. What about blood coming back to the heart? The pulmonary veins are going to bring oxygenated blood from the lungs into the left atrium, so the red side top, which is going to pump the blood into the left ventricle, as we've already seen, and that's going to be part of our pulmonary circuit. The superior vena cava, the inferior vena cava, and the coronary sinus are all going to be responsible for returning deoxygenated blood to the right atrium, the blue side up at the top, and that's part of our systemic circulatory system. So we're going to look at that pathway on a big chart in a few minutes, so don't get too panicky about it just yet. Let's talk about some other anatomy, and that's membranes of the heart. So the pericardium, or the pericardial sac, is actually made of 2 distinct sublayers. There's the outer fibrous pericardium and the inner serous pericardium. The fibrous pericardium, so if you look at your picture, it's the gray on the outside there. It's made of dense connective tissue. It protects the heart, and it maintains the heart's position in the thorax. The serous pericardium is the delicate 2-layered membrane. So the serous pericardium is going to be made of the parietal pericardium and then the visceral pericardium. The parietal pericardium is going to be fused to the inside of the fibrous pericardium. The visceral pericardium is also known as the epicardium and is fused to the heart. And it is also considered part of the heart wall. And then the space that's in between them is going to be filled with lubricating serous fluid that lies between the epicardium and the pericardium. So let's take a look at our picture. The first pink that you see over on the right-hand side that's closest to the fibrous pericardium, the gray, is going to be the parietal layer of the serous pericardium. And then the kind of purpley lilac one is going to be the visceral layer of the serous pericardium. You should have covered visceral and parietal membranes in 210. If you did not, they are at the end of my chapter 1 lecture on YouTube. The space in between them that's blue, that's the cavity where that fluid is. And that's where that lubricating fluid is. It's allowing those two membranes to slide past each other. When they don't slide correctly, it creates a condition called pericarditis. So now we've looked at the stuff that's on the outside of the heart. Now let's look at the actual layers of the heart wall. Remember, the heart itself is a hollow structure, and it is got a wall, and that wall is 3 layers thick. The outermost one that I just talked about is called the pericardium. The outermost one is called the epicardium. Sorry, I can't talk today. Or the visceral pericardium. That's that pink one, right? The middle and the thickest layer, that's bright red or the red color there, that's made of cardiac muscle cells that you learned about in Bio 210 as well, and that's your myocardium. Remember, MY means muscle, so that's the muscle layer. And then the black line that you see is a single layer of cells called the epicardium, and that is the lining of the chambers and covers the heart valves. It's going to become continuous with the vessels that we'll be learning about in the next chapter. So we've got those 3 layers of the heart wall. The largest one, of course, is the myocardium, and that's the one that's actually doing the job of pumping the blood out to the heart, out of the heart, and out to the major arteries. I already mentioned the idea that you've got 2 chambers, you've got 4 chambers, you've got 2 atria and 2 ventricles. What's separating those? You have an interatrial septum, so between the 2 atria. You have an interventricular septum, which means between the 2 ventricles. And then you have an atrioventricular septum, which is the entire structure separating both sides. In our picture, we can't see our interatrial septum. It's hiding back behind that pulmonary trunk. So you see the pulmonary trunk, which is blue. They've got it opened up so you can see the pulmonary valve in there. If we were to strip that away, you'd see that wall continue like you see of the interventricular septum below it. Veins and arteries, I've been using those words a little bit already, so let's talk about them. So veins bring blood into the heart. So in veins should remind you that it is bringing it into the heart. Arteries, the capital A at the begin or the A at the begin, means that it takes blood away from the heart. So one thing I need for you to unlearn, if you've learned it this way, is that arteries carry oxygenated blood and veins carry deoxygenated blood. That is not always true. That is not true in the pulmonary circulatory route, and it's not true in fetal circulation. So in general, that's true. And when we talk about systemic vessels in the next chapter and in lab, that's mostly what we're looking at. And yes, if it's blue, it's a vein, and if it's red, it's an artery. But in this chapter, that's not the case because we have pulmonary vessels that we're going to be looking at. So our pulmonary artery is actually blue, and our pulmonary vein is actually red because they are carrying blood to and from the lungs, and we don't name them according to the oxygen level in the vessel. We name it according to the direction that it's making the blood move. So I had mentioned the idea that superior and inferior vena cava bring deoxygenated blood to the right atrium from all over the body. There's also another structure that I mentioned that I want to talk about briefly, and that's called the coronary sinus. The heart is a muscle, and just like any other muscle in your body, it has to have a blood supply. And so we're going to learn about the coronary arteries that take blood out to the heart itself, and that blood is returned to the circulatory system as well, and it's returned in what's called a structure called the coronary sinus. So that's going to return blood into that right atrium along with the superior and inferior vena cava. The pulmonary veins are going to bring oxygenated blood from the lungs to the left atrium. So the pulmonary trunk sitting right on the very front of the heart, and it's going to carry the blood from the right ventricle to the lungs for reoxygenation. So we just saw blood coming in through the superior vena cava up at the top, the inferior vena cava at the bottom. There's little white arrows there, and the coronary sinus that you can't see on this picture. I have another picture of it coming up. It's going to go from the right atrium down to the right ventricle, and then it's going to head up into the pulmonary trunk. And let's look at the other side, the red side. The aorta is going to carry the oxygenated blood that came in from the right atrium into the the left atrium into the left ventricle into the aorta. So the myocardium is going to have some differences. The myocardium in the left ventricle, because it's taking blood out to the entire body, is going to be significantly thicker. So both ventricles are pumping the same amount of blood, but the left ventricle has to generate a whole lot more pressure to overcome the resistance that it's going to encounter in the vessels. So looking at our picture, remember the right-hand side is the side farthest away from the lettering there, and the left-hand side is the side closest to all the words that I have on the screen. So look at that myocardial layer. Notice how it's much thicker in that left ventricle compared to what you see in that right ventricle. A little bit about valves. So we're going to see valves in vessels as well, and we're going to see valves other places in the body. Valves are always about preventing backflow. Always about preventing backflow. So they are one-way valves. They keep things moving one direction. In this case, what we're moving in one direction is blood. We'll see that we have valves in the digestive system to keep things from going the wrong way in the digestive system. We have it in the urinary system. We have them lots of places. I think I read somewhere that you have like 200 valves in the body. I don't know if that's correct or not, but I read that somewhere a long time ago. So yeah, they're all about making sure that nothing goes the wrong direction. We have two types of valves in the heart that we're interested in. We have a valve that's sitting between the atria and the ventricle on both sides, and that's called the atrioventricular valve. And you're going to have a right and a left of those. And so when you're looking at the picture, they're white, and they look like they've got like strings attached to them. Those are your AV valves. Then you're going to have a pulmonary and aortic semilunar valve that's going to be found between the ventricle and the artery that it's dumping blood out into. So when you look at the pulmonary trunk, you see a white valve in there. That's going to be your pulmonary valve. And then you're going to see sitting next to that, it's kind of hard to see because it's back behind there, is your aortic valve. So again, the valve between the atria and the ventricles is called the atrioventricular valve. It has some alternative names. So on the right-hand side, you can call it the right atrioventricular valve, or you can also call it the tricuspid. And it's called the tricuspid because if you look at it, it's kind of the pale blue structure we're looking at there. It has three cusps, right? Three flaps. The other one on the other side is called the bicuspid or the mitral valve. That's your left. This is all your left atrioventricular valve. So bicuspid, mitral valve, or left atrioventricular valve all mean the same thing. And it's again between the left atria and ventricle. So if you're trying to remember which one comes first, we always go from right to left whenever we're looking at things in anatomy. So when we go from right, left, it's tricycle before bicycle. You ride a tricycle before you ride a bicycle, right? So tri is on the right side, bi is on the left. All right, the other two sets of valves. So if you look in the middle on our picture, you see one that's in the aorta and one that's in the pulmonary trunk. So red and blue below that. That's the ones I'm talking about there. Those are called the semilunar valves. They're called semilunar because they look like three half moons. The pulmonary semilunar valve is going to be found between the right ventricle and the pulmonary trunk. It has some alternative names as well. So it can be called the pulmonic valve or simply the pulmonary valve. So you can take the word semilunar out. The aortic semilunar valve is located between the left ventricle and the ascending portion of the aorta, the first portion of the aorta, and it's known as the aortic valve. It doesn't have any other name other than aortic valve or aortic semilunar valve. The atrioventricular valves, the ones that are between the atrium and the ventricle, they're attached to the walls of the ventricles by strong cords of connective tissue that are called the chordae tendineae. The chordae tendineae are connected to the papillary muscles in the wall of the ventricle. So let's take a look at that. So you see what looks like little spider webs. Those are the chordae tendineae. And notice that they're attached to these out projections of muscle. So a papilla is a finger-like projection of something. It's usually muscle, but not always. But it's a finger-like projection. In this case, it is a finger-like projection of muscle that just literally sticks into the inside or the inside of the chamber of the ventricular chambers. The chordae tendineae are guiding the cusps of the valve into the closed position, and they prevent them from everting or flipping into the atria. They do not assist in opening the valves. So when we talk about the valves opening, these guys don't pull on them and open them. All they do is just make sure that they close properly. They remind me of if you've ever watched the Macy's Thanksgiving Day Parade and you see the balloon handlers. The balloon handlers, basically what they're doing is they're just guiding those balloons down the road and making sure that they don't go the wrong direction or any of those kinds of things. So that's kind of what these chordae tendineae are doing. They're just guiding these cusps to the valves, the flaps to the valves, and making sure that they go back into position correctly. Both our AV valves open at the same time as the blood pressure builds up in the atria. So that's what opens them, not those chordae tendineae. So as the blood pressure fills up in that atria and starts to exert pressure on them, that's what causes them to open. And once they are open, they allow the blood to pass into the ventricles. The semilunar valves are open at the same time as well, and that is due to the pressure from the blood in the ventricles. So the pressure is a result of the contraction of the ventricle. So as the ventricle contracts, it puts pressure on the inside of that closed valve and causes it to open. This pressure also closes the AV valves from the inside, which is what's preventing the backflow of blood into the atria. All right, so here's the rule. When one set of valves is open, the other set is closed. And when I say a set, I mean the AV valves. So the right and left AV valves or tricuspid and bicuspid, that's considered a set. And the pulmonic and aortic valve, that's considered a set. So when one set is open, the other set is closed and vice versa. What are some of the disorders of heart valves? So when they don't function properly, they are described as being incompetent, and they can result in valvular heart disease. So valvular heart disease can range from something that's benign, meaning that no problem whatsoever, or it can be lethal. A prolapsed valve occurs when one of the AV valve cusps that I was just talking about don't close properly. The most common form of this is mitral valve prolapse. You may have heard of that before. That results in a heart murmur. That's over on the left-hand side between the top and the bottom, and you have some backflow into the right atrium. What that sounds like when you listen to it underneath of through a stethoscope is it sounds like a whooshing sound. So you'll hear that the traditional lub dub, lub dub, lub dub sound, but you'll hear lub whoosh dub, lub whoosh dub. So between the two heart sounds, you'll hear a whooshing. Stenosis is another relatively common condition that occurs when the heart valves become too rigid and they calcify over time. And so when you have stenotic valves, which is more common with the semilunars, but the stenotic valves are going to result in a high-pitched kind of squirrely sound in the stethoscope. All right, here is something you absolutely have to know. So remember in the endocrine chapter, I said there was a few things that when I spoke to nurses in nursing programs over the years, what would be the most beneficial things for students to know when they got to you that I could teach them? Well, one was all of that hormone stuff, and most particularly the renin, angiotensin, aldosterone system that we talked about. But another one was pathway of blood through the heart. So if you know this, this is going to be a night off for you when you go to study. All right, so we're going to learn this. You're going to know this really, really well. So I want you to notice, first of all, that I've color coded it for you. So the right side is blue because that's representing the deoxygenated side of the heart. And then the left side is red, and it is denoting the oxygenated side. All right, so we're going to start at where blood is returned to the right-hand side and trace it all the way through. All right. So blood returns to your heart from the superior vena cava, the inferior vena cava, and the coronary sinus. From there, it goes to the right atrium. It passes through the tricuspid valve or the right atrioventricular valve into the right ventricle. It then passes through the pulmonary semilunar valve into the pulmonary trunk. The pulmonary trunk is going to split and become the right and left pulmonary arteries. And then it goes to the lungs where it reoxygenates. So once blood is reoxygenated, it's going to head back to the heart, and it's going to do that via the right and left pulmonary veins, and there are actually 4 of those. Into the left atrium, through the mitral valve or bicuspid or left atrioventricular valve, whichever term you like to use, into the left ventricle, through the aortic semilunar valve, into the ascending aorta, which is the first portion going up, into the aortic arch, which is the curved part, and then into the descending aorta, which is the part that drops straight down and out to the rest of the body. So you might be saying to yourself, why does it matter if we say aortic, ascending, the arch, or the descending portion? Because there's vessels that come off those first 2, and we're going to learn those here pretty quick. So we want to make sure that you know where those are coming from. So make sure you separate that aorta into 3 different parts. So there it is in a picture, all of those structures that I've just talked about, right? And arrows. You should know this so well that you can pick it up anywhere in that circulatory route and tell me the next steps. And you'll have questions on your quiz and questions on the exam that say, from here, blood goes here next, and you've got to fill in those blanks. All right, so make sure that you know this blood flow through the heart. There's a whole bunch of really cute things on YouTube. I don't know if it's still on there, and maybe it's not, but it was for the longest time. It was children, and I think they were maybe in India or Pakistan, and they were out in a parking lot, and they had drawn the heart on in chalk on the parking lot, and they had the kids in red and blue shirts, and they were walking the pathway through the heart, and as they switched from deoxygenated to oxygenated, they changed their shirts. So they had like 2 shirts on, one on top of the other, and they kept putting one on and taking one off. And it was just the most adorable thing to see these kids doing that. And they were saying it as they were going through, which was really fascinating. Um, I've seen people on there that write songs, write poetry. I've seen a rap song on there on Blood Through the Heart. However, you need to learn it. You need to make sure that you learn it. So here's an opportunity for you to use your own creativity and figure out a way to learn blood flow through the heart that is going to be beneficial to you in the long run. So find a way that speaks to you, and spend a little bit of time learning this pathway because it's critically important. There are some other circulatory routes that we need to talk about. The coronary circulatory route is a short circulatory pathway that delivers blood to the actual heart muscle, the myocardium itself, and other components of the heart. It is not continuous. It is rather it cycles and it peaks, reaches a peak when the heart muscle is relaxed and nearly ceases when it's contracting. So it's like it's pulsing with the heartbeat itself. So what is this pathway? So coming up from the out of the left ventricle, the ascending aorta, that first little chunk of aorta that comes up, there are two small vessels that branch right off of that and out to the heart, and they are called the right and left coronary arteries. All right, so the right and left coronary arteries. And then they're going to branch, and we're going to see this over in the lab. So it's hard to see this without a picture, but the branches from each artery are going to include the circumflex that's going to branch into something called the marginal, and then the one that's coming, those are coming off the right-hand side, and the one that's coming off the left-hand side is going to go straight down the front of the heart and become the anterior interventricular artery, also known as the anterior descending. The right coronary artery is going to distribute blood to the right atrium, portions of both ventricles, and the heart conduction system that we'll be talking about. And it also originates from the ascending aorta, as I mentioned. The two branches of this artery are going to include the marginal arteries that supply blood to the superficial portion of the right ventricle, and then the posterior interventricular, which is going down the back side of the heart, also known as the posterior descending, that's going to supply the interventricular septum and portions of both ventricles on the back side. Whew, pictures help so much. So I want you to look in the very center, and you see the ascending aorta. They don't have it labeled, they just have it labeled aorta. You see that? And you see the two vessels that are coming off of that ascending aorta pretty early on, right after that valve. So the thing that kind of looks like a hiney is your semilunar valve. Um, so right above that, you have two little vessels coming off of that. When you see them faded like that, that means they're behind another structure. Okay, and so those are your right and left coronary arteries. So let's look at the left one first. The left one first. So the left one is going to come out, and it's underneath of the auricle, and then it's going to dip down and go, one branch is going to go around the side called the circumflex. All right, so it's going to go around, it's going to circumnavigate, it's going to go around towards the back, and there's a left marginal that's going to come off of that and run down the left-hand side of the heart. The one that's really important to talk about, though, is that one that goes straight down the middle there. That's called the left anterior descending or interventricular artery, and that is the one that is delivering blood to the left ventricle. And of course, the left ventricle is the one with the thickest wall and the one that's pumping blood out to the whole body. If someone experiences a infarction, a myocardial infarction, what we call a heart attack, anywhere in there, it can be life ending. All right. So the further up that vessel, the closer to the coronary artery that event occurs, the less likely your patient is to live, because it's preventing blood from going anywhere downstream. That's the one that they call the widowmaker. So if you've heard that term before, that somebody had a heart attack, and it was in the widowmaker vessel, that's the vessel that they're talking about right there. Let's look at the right-hand side. So again, you see the right coronary artery, and it just rolls around towards the back, kind of runs along that that auricle that's up there, that flap that you see, and rolls around. And then there's a right marginal artery that goes down along the side of the heart on the right-hand side, but mostly that right coronary artery just goes around, and it goes around to the back, and notice that it's faded, and then you've got a vessel that runs right down the back just like you had going down the front. That's called the posterior interventricular or the posterior descending artery. So coronary veins are going to drain the deoxygenated blood from the heart muscle that was delivered by the coronary arteries. The great cardiac vein can be seen on the front surface, and it follows along the interventricular sulcus. So where the anterior descending and the great cardiac sit, there's kind of a little groove, and that's called the interventricular sulcus. And so it's going to be sitting in there as well, and it's going to be draining the areas that were supplied by that anterior interventricular artery. It receives deoxygenated blood from several major branches, so they're all ultimately merging back into that great cardiac vein. So it's going to get blood from the posterior cardiac vein that drains the area supplied by the marginal artery, the middle cardiac vein that's going to drain the areas supplied by the posterior interventricular artery, and the small cardiac vein that drains blood from the posterior surface of the right atrium and the ventricle. All of those veins, including the great cardiac vein, are ultimately going to merge into the back of the heart into a flattened vessel called the coronary sinus. So this is a large flat vein on the back side of the heart that empties directly into the right atrium from the back side. So that was the third way that blood returns to the heart, by the coronary sinus. The anterior cardiac vein drains the anterior surface of the right ventricle, and it drains directly into the right atrium rather than having to go into the coronary sinus. All right, so looking at the front side of the heart, you can see the red vessels that we were already talking about, and you can also see the blue ones. So the great cardiac vein is sitting there right next to the anterior interventricular artery, and it's taking blood. If you follow it up, and then you see that it goes to the right, and it travels along with the circumflex, and it goes around to the back where it'll go into the coronary sinus. There's the back side of the heart. So that big blue vessel that you see that's sitting in the sulcus, that's between the atria and the ventricles, that is your, once it rolls around to the back, that's called the coronary sinus. And notice that it just kind of like disappears into the back of the heart, and it actually has an opening that dumps the blood right into the right atrium. You can also see, let's see, what else can you see on this picture? You can see the middle cardiac vein down at the bottom. That's again draining the blood that's coming from the posterior descending. So you'll be looking at those vessels in a little bit more detail in lab. Some disorders associated with the coronary circulation, one of them is atherosclerosis. This is hardening of the arteries. You have an accumulation that's called a plaque that forms in the lumen, or the opening in the middle of the vessel, that includes fatty material from cholesterol. Then you also have connective tissue in there, some white blood cells, some smooth blood muscle cells, just all kinds of stuff sort of build up and make this blockage. Blood is restricted because of it. That condition is called ischemia. It causes the cells in the area to receive insufficient amounts of oxygen and results in hypoxia. Without a supply of oxygen, the cardiocytes may die, resulting in a myocardial infarction or an MI or a heart attack. So unlike skeletal muscle that could be without oxygen and continue to operate, what we called anaerobic metabolism, and you ended up with a buildup of lactic acid, and that created the muscle soreness, et cetera, et cetera, et cetera. Heart muscle doesn't work that way. Heart muscle is only aerobic, and so these buildups that happen can lead to all kinds of issues. So a little more detail about cardiac muscle cells. So cardiac muscle cells, if you remember from, I think it's chapter 9 in 210, they are short branched striated cells with a single centrally located nucleus. Occasionally they have a second nucleus, but typically just one. Cardiac muscle cells are joined to each other by intercalated discs. So intercalated discs are going to connect the cardiocytes and support the synchronized contraction of the heart muscle, what is called functional succinctum. And that's what we want. We want every single muscle in that particular chamber, every cell in that chamber, to be contracting at the same time. That was not what we wanted with skeletal muscle, right? But in heart muscle, we do. The discs that connect these muscle cells to each other are made of desmosomes, tight junctions, and gap junctions, and they allow ions to pass between the cells, and they help to synchronize the contraction. If you don't remember all those kinds of junctions, please go back to chapter 1 in my YouTube lectures. The importance of this is all of the cells are all bound together, and that's going to allow for a greater force of contraction. So looking at it underneath of the microscope, they're pointing out the intercalated discs. The intercalated discs to me make cardiac muscle look like bamboo. I always see it, and that's the first thing that I think of. And then they're pointing out the nucleus as well. Cardiac muscle cells contract without stimulation from the nervous system. So your nervous system has the ability to influence the rate of contraction and the force of contraction, and we'll talk about how that happens. But the actual generation of the action potential is not through our nervous system. You have special cells called nodal cells that are going to generate that electrical signal. The pathway of cardiac conduction is the sinoatrial node, or the SA node, the AV node, or the atrioventricular node, the atrioventricular bundle, the right and left atrioventricular bundle branches, and the Purkinje fibers. So another pathway that you have got to learn. But this is a shorty. It's only 5 steps. The total time elapsed from the initiation of the impulse in the SA node until depolarization of the ventricles is approximately 225 milliseconds. All right, so let's look at the pathway of this conduction system. Look up in the roof of the right atrium and you see the SA node, or sinoatrial node. That's a special complex of cells that can generate an action potential. And so that electrical signal is going to travel down the walls of the right atrium down to the floor of the right atrium to the AV node. Then that signal is going to pass over to the atrium, or to the septum, and the portion that is traveling from the AV node over to the septum is called the atrioventricular bundle. The old name for this was the bundle of His, like a snake hiss, but spelled with a capital H and only one S because it's somebody's name. Either way is fine. Atrioventricular bundle or bundle of His is fine, either term. And then it's going to split and run down either side of the septum, and those are called the right and left bundle branches. And then each of those bundle branches is going to wrap around the apex of the heart and start to produce what are called the Purkinje fibers. And it's the Purkinje fibers that are going to deliver the electrical signal and zap the heart and cause it to contract. All right, a little bit more detail about each of these different steps. So the sinoatrial, or SA node, is what establishes the normal cardiac rhythm, or what we call the normal sinus rhythm. It's located in the superior and posterior walls of the right atrium. I would just say the roof of the right atrium to get the idea. It has the highest inherent rate of depolarization and is known as your pacemaker. And so it is producing electrical signals at a rate of about 100 times a minute. It initiates the sinus rhythm, or the normal electrical pattern, that's followed by the contraction of the heart. Then we move down to the floor of the right atrium and we find the AV node, or atrioventricular node. This is a smaller diameter of cells in the node that delays the transmission impulses. So signals take about 100 milliseconds to pass through this node. This allows the atria to contract at the same time and complete its contraction before the impulses are transmitted to the ventricles. The AV node has an inherent rate of about 50 beats per minute in absence of the SA node. So what that means is if your SA node stops working, your AV node can take over, but there's a trade-off, and that trade-off is that you're only getting about 100 beats per minute. Then we go to the atrioventricular bundle, or the bundle of His. This is located at the top of the interventricular septum. It connects the atria to the ventricles electrically. And then we split into the right and left bundle branches. The left bundle branch is going to supply the left ventricle, and the right bundle branch is supplying the right ventricle. And that's going to carry the signal towards the apex, or the pointy part of the heart. And then finally, we get to the Purkinje fibers. These are what wrap up around the outside wall of the heart on each side. They extend through the myocardium from the apex of the heart toward the atrioventricular septum and the base of the heart. Since the electrical stimulus begins at the apex, the contraction also begins at the apex and travels towards the base of the heart. This allows blood to be pumped out of the ventricles and into the aorta and the pulmonary trunk. So there it is. If you start over on the left-hand side, they're showing you in purple what's contracting. So we start with the AV node or the SA node, and it travels to the AV node, and what that does is cause the atria to contract. And then we have the contraction that's happened. We're passing that signal, not the contraction, we're passing the signal down the septum at this point, and we cause the Purkinje fibers to deliver the electrical signal to the ventricles and cause the ventricles to contract, and then further into that contraction as it starts to go all the way up to the heart, and we start all over again. So I mentioned that the nervous system doesn't generate the electrical signal of the heart. It only influences the heart rate and force of contraction from two cardiovascular centers in the medulla oblongata. The acceleratory center is what causes an increase in the heart rate and force of contraction via your sympathetic stimulation. So you guys have all had this happen before. You're in a fight or flight situation, whatever that is, and all of a sudden your heart starts beating much faster, right? And that's because of the adrenaline that is running through the sympathetic side of your nervous system. The cardioinhibitory center is going to decrease the heart activity via the parasympathetic stimulation of the vagus nerve, which was cranial nerve number 10. Cranial nerve number 10. If you did not learn all your cranial nerves, I'm going to send you back to Bio 210 again and look that up. I don't remember which chapter it is. I want to say it's either 10 or 11. The vagus nerve sends branches to both the SA and the AV node and to portions of both the atria and the ventricles. During rest, both centers, the cardioacceleratory and the inhibitory, stimulate the heart, and that creates what's called autonomic tone. So we had muscle tone in our skeletal muscles as well. We have the same thing going on with the heart. Normally, vagal stimulation is what's in charge. If it's left unregulated, the SA node would initiate a sinus rhythm approximately 100 beats per minute, which is way too many. At rest, the heart beats at about 60 to 100 beats per minute. Some disorders associated with the heart, some arrhythmias. These include irregular heart rhythms. These result in an uncontrolled, uncoordinated atrial and ventricular contraction. So what should be happening is the top chambers are going to contract, and then the bottom chambers contract, and then the top chambers contract and the bottom chambers contract. That's how it should be going, right? So if we have irregular rhythms, then those two things aren't happening synchronously. Bradycardia is a condition in which the resting rate is below 60 beats per minute. So your patient is sitting there, you take their pulse rate, and it's 55. All right. And that's absolutely fine if you're talking about a well-trained athlete. So if you're a person sitting there, runs marathons and, does all kinds of athletic type stuff, it's obvious that they're physically fit. Bradycardia is very common, right? So that is typical for them. If your patient is sitting there and they weigh 350 pounds and they smoke 2 packs of cigarettes a day and they have a heart rate at rest at 60 beats per minute, something's not right. Okay. Tachycardia is when the resting heart rate is above 100 beats per minute. So they're sitting there, and everybody's a little bit nervous when they're, getting stuff like that done. So it'll be a little elevated, but they're sitting there just minding their own business, not doing anything, and their heart rate is over 100 beats per minute. The normal heart rates in children and in adults during exercise are often above 100 beats per minute. We expect to see that. So now let's switch gears and let's talk about how action potentials occur in our cardiac contractile cells, or our nodal cells, some books call them. This is a rapid depolarization followed by a plateau phase and then repolarization. So I want you to think back for a second to nervous system. I think that was chapter 11 in 210. And I want you to remember how it was basically a great big spike that happened. So you had your action potential, you had your resting membrane potential, and then it started to rise, and then you had that great big spike, and then it came back down, right? And then it reset itself. A little bit different when we're talking about what we see with the heart. So over on the left-hand side, you see the rapid depolarization in red. And then rather than a spike that drops back down, you see this plateau where it sits there for a second, and then it starts to repolarize. So a very different shaped graph that we see in heart. This phenomena accounts for the long refractory period that's required for cardiac muscle cells to pump the blood efficiently. So what are the steps in the action potential in cardiac contractile cells or pacemaker cells? So we have the voltage gated channels that rapidly open at the beginning of the depolarization. The membrane potential is going to go up to positive 30 millivolts. So that number is basically the same. Remember, we start around negative 70. In the case of the heart, we start around negative 90 usually, but we go up to positive 30, and it's at that point the sodium channels are going to close. This depolarization period is very rapid and short. So that's the red on the chart that you're seeing. All right, next we have the plateau phase. And so you see that kind of purple sort of flat line. This is a slow decline that you're seeing. This is due in large part to the opening of the slow calcium channels. This allows calcium ions to enter the cell while few potassium channels are open, and allowing the potassium to exit the cell. The relatively long plateau phase lasts approximately 175 milliseconds. That plateau sustains contraction for the longer time period, and we want that. This is needed to ensure that we have a powerful contraction that empties the ventricles and pushes the blood into the aorta. Once the membrane potential reaches zero, the calcium channels are going to close and the potassium channels are going to open, which is going to allow the potassium to enter and to exit the cell. The repolarization lasts approximately 75 milliseconds. This is the blue part of the graph that you're looking at. And at this point, the membrane potential drops until it reaches the resting level and the cycle repeats. This entire event lasts between 250 and 300 milliseconds. The absolute refractory period for cardiac contractile muscle lasts approximately 200 milliseconds, and the relative refractory period lasts approximately 50 milliseconds. That's different than what we saw with skeletal muscle. During the absolute refractory period, another electrical signal cannot be generated. Without extended refractory periods in cardiac muscle, premature or weak contractions would occur and lead to insufficient blood supplies. So we would have blood being ejected out before it had a chance to completely reset itself. All right, the differences between skeletal muscle contractions and cardiac muscle contractions. The action potential in skeletal muscle lasts between 1 and 2 seconds. In cardiac muscle, it lasts 200 milliseconds. So way different action potential timing. And then the contraction in skeletal muscle lasts between 15 and 100 milliseconds, whereas in cardiac muscle, the contraction lasts over 200 milliseconds. What's the benefit of the longer action potential and contraction? We want that sustained contraction. It ensures efficient ejection of blood from the heart. The longer refractory period is going to prevent tectonic contractions. You saw those in skeletal muscle, and you actually want those skeletal muscle. That's what allows you to have sustained contraction of, like, let's just say, to hold up a book or whatever it is that you're holding up or you're doing or you're lifting or you're moving or however it is you're doing that. In heart, that's not something we want. So some really significant differences between those two. And then just looking at that graphically, again, the action potential versus the contraction in skeletal and cardiac muscle. All right, let's move along to EKGs or ECGs, the electrocardiograms. These are something that's done routinely in a lot of situations. So most of you will learn very quickly how to do these and do plenty of them. It is a tracing of the electrical signal of the heart, and it reveals a detailed picture of both normal and abnormal heart function and is an indispensable clinical diagnostic tool. The standard EKG, the electrocardiograph, which is the machine that generates the EKG, uses 3, 5, or 12 leads. The greater the number of leads it has, the more information it's going to provide. So here's what it looks like. It records the complete electrical activity of the heart, not a single action potential. So we're looking at all of the activity that's going on. An ECG appears as a series of waves, or deflections is another term for that, with 5 prominent points: the P wave, the QRS complex, and the T wave. Each point indicates a certain activity at a certain place in the heart. The P wave, which is the first little bump that you see over there, represents the depolarization of the atria. So depolarization and the word contraction mean the same thing. All right, depolarization and contraction mean the same thing. The atria begin contracting at approximately 25 milliseconds after the start of the P wave. We have the PR interval, which is along the bottom on our EKG on the side. It is the period between the start of atrial excitation to the start of the ventricular excitation. The large spike, what they call the QRS complex, represents depolarization, again contraction, of the ventricles, and at the same time it is representing the atrial repolarization or relaxation. So it's actually representing two things. The ventricles require a stronger electrical signal because of their larger size. The ventricles begin to contract as the QRS reaches the peak of the R portion of it. So you can see QRS. So Q is down at the bottom, at the base. R is a spike, and then S is back when it returns. The T wave represents ventricular repolarization. So repolarization and relaxation mean the same thing. So R and R. The QT interval is the period between ventricular depolarization and repolarization. The ST segment is the period of complete ventricular depolarization or contraction. The PR segment is the time delay between the atrial repolarization or relaxation and ventricular depolarization or relaxation, or contraction. It reflects the delay that we see at the AV node. The TP, so the end, all the way to the beginning of the next one, is a complete depolarization of the entire heart, and there's no electrical activity. So taking a look at this, you can see over on the left-hand side, it shows you where to start, and it shows you on the EKG what part we're looking at each time. So you can see how each one of those, how the EKG corresponds to what we're seeing with the heart. The cardiac cycle is the period of time that begins with the contraction of the atria and ends with the end of ventricular relaxation up to the beginning of the next atrial contraction. So it's not just the contraction of the atria to the repolarization of the ventricles, it's the complete repolarization of the ventricles in that last period that we talked about, that weird time period where there's no electrical activity that goes all the way through that. So the cardiac cycle is one complete cycle when we start all over again. Systole is the period of contraction that the heart undergoes when it pumps blood into circulation. Diastole is the period of relaxation that occurs as the chambers are going to fill with blood. Both the atria and the ventricle undergo systole and diastole in an alternating fashion. So the top, then the bottom, then the top, then the bottom. So when the top is contracting, the bottom is relaxing. And when the bottom is relaxing, the top is contracting, right? So taking a look at this cardiac cycle, the cardiac cycle begins with atrial systole and progresses to ventricular systole, atrial diastole, ventricular diastole. And when ventricular diastole is done and we're ready to start with another atrial systole, that's one complete cardiac cycle. We can divide that into 3 major steps: ventricular filling, ventricular systole, and isovolumetric relaxation. Ventricular filling is when both the atria and the ventricles are in diastole. Both the blood is going to passively flow to the atria, and the blood pressure is going to open the AV valves, and the blood will flow into the ventricle. So literally, blood is just coming into the heart and falling through the atria into the ventricles. The P wave is going to occur, and it initiates the atrial contraction, which forces whatever blood that is left in the atria into the ventricle. So it squeezes that last little bout of blood in, and at the same time, the ventricles start to depolarize, which is the beginning of the QRS complex. Early ventricular systole, the atria are in diastole and relaxed and almost empty. The ventricles depolarize and begin to contract. The AV valves are going to close because of the pressure that builds in the ventricles. Now, the AV valves are closed, and for a brief period of time, so are the semilunar valves. And that period where both of those are closed is called isovolumetric contraction. This is when we're building up the pressure in the ventricles. Late ventricular systole, the sustained contraction of the ventricles increases the blood pressure within the ventricles. Once this pressure exceeds the pressure that's in the arteries, the pulmonary trunk, and the aorta, the semilunar valves open. So how do those valves open? It's because of pressure changes. So when the pressure in the ventricles gets higher than the pressure in the vessels, it causes those semilunars to open up. That's why high blood pressure is so dangerous. Because when you have high blood pressure, the ventricles have to get to an even higher pressure to push that blood through. Blood flows from the ventricles into the great vessels, and this subphase is called the ejection phase. Some blood will remain in the ventricles. This is called end-systolic volume, or ESV. And then finally, the isovolumetric relaxation. Both the atria and the ventricles are in diastole. The ventricles begin to repolarize, which is going to produce that T wave. The atria begin filling with blood. The AV valves are closed. The SL valves are closed due to to keep the blood that just was ejected from backflowing. Once the blood pressure in the atria opens the AV valves again, the cycle starts over. So I really like this picture. This is representing the relationship between the cardiac cycle, which is down at the bottom, and an EKG, which is the squiggly line you see at the top. So you can look at that. You can see that initially both the atria and the ventricles are relaxed, which means they're in diastole. Then we have the P wave, which represents the depolarization of the atria, which is going to lead to the atrial depolarization or contraction in systole. The atrial systole is going to extend until the beginning of the big spike, the QRS complex, and then they relax during that process. The QRS complex is representing the ventricle depolarization, which is followed by ventricular contraction. We have to have a depolarization before we can have a contraction. And then the T wave at the end represents the ventricle repolarization and marks the beginning of the ventricular relaxation. Heart sounds. So in a normal, healthy heart, there are two audible heart sounds, S1 and S2, both caused by the closing of the valve. So what you're listening to is the closing of the two different sets of valves. S1 is the sound created by the closing of the AV valves during ventricular contraction, and it is normally described as LUB, L-U-B, or the first heart sound. S2 is the sound of the closing of the semilunar valves during ventricular diastole, and it is described as a dub sound. The silence between the two of them refers to heart relaxation. The mitral valves close before the tricuspid valve, and the aortic valve closes slightly before the pulmonary valve. If you're not familiar with this sound, YouTube is your friend. Go listen to it on there, and you really can hear the distinct difference between those two. So make sure you spend a little time listening to that. Again, the mitral valve is going to close before the tricuspid, and the aortic valve closes before the pulmonary. When I say before, I just mean a second before. I mean a tiny fraction of a second before that. Each valve can be heard separately if the stethoscope is placed in different positions on the body. So you may have noticed a physician, when they're listening to someone's heart, they're moving that stethoscope around. And what they're doing is listening to those different valves. Cardiac output. So cardiac output is a measurement of the amount of blood pumped by each ventricle in 1 minute. The equation for that is heart rate times stroke volume, HR times SV. Heart rate is the number of beats per minute. All right, we know how to measure that. Stroke volume is the volume of blood pumped by one ventricle with each beat, right? And your heart beats 70 times a minute, right? In a normal healthy heart, that's roughly 5.25 liters per minute. That's how much blood is pumped through the heart each minute. So go back to my 2-liter Coke bottle analogy and realize that this is 2.5 roughly, or 2 1/4 bottles of Coke, right? There's lots of factors that can influence cardiac output. All right, so here are some of the factors that can affect your cardiac output. So things that affect the heart rate include autonomic innervation, your fight or flight, and your parasympathetic side. Hormones, fitness level, age, those are all things that are going to factor into what an individual's heart rate is. And then factors that affect the stroke volume include the heart size, fitness level, gender, contractility, duration of contraction, and preload and afterload. So preload is how stretchy the heart muscle is and how easily it can go back to what it was before. And then afterload is how, I mentioned this already, afterload is how much pressure do we have to put on the ventricles to get the pressure in the ventricle higher than what's in the vessel, so that blood will move, so that the valve will open and blood will move. So those are important things to understand too. So stroke volume is calculated by your end diastolic volume minus your end systolic volume. So those are measurements that they can get. So those two things, the heart rate times the stroke volume, is where we come up with that 5.25 liters for the average patient. All right, a little bit more detail about preload and afterload. So preload is the volume of blood that's in the ventricles at the end of diastole. So what they call the end diastolic pressure. This is hypervolemia, regurgitation of the cardiac valves, heart failure. Afterload is the resistance that the left ventricle has to overcome to circulate the blood. So as I said in the previous slide, remember that we're building up pressure, and the pressure in the ventricles has to get higher than the pressure that's in the vessel to cause those valves to open, the semilunar valves to open, and for blood to flow. So that's what afterload is. Afterload is increased by hypertension and vasoconstriction, which is another thing we had talked about earlier. So increase in afterload equals an increase in the cardiac workload. So the last little part that we talk about here is fetal heart development. So the fetal heart is going to develop through a very complex, well-orchestrated process that begins about the 3rd week of gestation, transforming it from a very simple tube into a 4-chambered organ that's capable of circulating blood before birth. So it's a misnomer. A lot of people think that mom's blood is circulating through the baby. That is not what is happening. So there it is. It starts out as just this 2 little tubes, and they join together and they form the top and the bottom, and then they turn into what looks kind of like a snail, and they continue to develop, and then we have a 4-chambered heart early in development. Now, mom's blood is not passing through the baby. She's exchanging with the baby through the placenta. And because the baby is not breathing yet, then mom's blood has to oxygenate the baby's blood through the placenta, just like mom has to feed the baby through the placenta. So there's some adaptations in the circulatory system of a fetus compared to a newborn baby. And so the fetal heart is going to support circulation because it has these specialized features, or what they call shunts. And the first one is called the foramen ovale. So between the right and left atria, there is an opening. And that opening allows blood that's coming into the heart to pass directly over to the systemic side. So normally blood comes into the heart, it goes through the pulmonary circuit, it comes back to the heart, goes out to the systemic. Well, you can't go out to the pulmonary circuit in a fetus because they're not breathing yet. So the blood comes in already oxygenated. And if that blood comes in oxygenated, there's not really any reason for it to go through the pulmonary side because it's going to go out to the lungs and there's nothing to do out there, right? There's no exchange to happen. Now, there's a small amount that needs to go out there to make sure that it's maintaining the tissues, but there's no exchange that's going on there. So that shunt, that opening between the right and left atria is called the foramen ovale. Once the baby is born and takes that first few breaths, that opening closes and becomes the fossa ovalis. So a foramen is a hole. A fossa is an indention. If that doesn't happen, they have to go in and close that. The ductus arteriosus, this links the pulmonary artery to the aorta. The pulmonary artery to the aorta. So when the pulmonary trunk comes out of the heart, it splits immediately into the pulmonary artery. Again, remember, there's no need for blood to go out to the pulmonary side. So even if blood tries to go out there, there's a bypass that puts it back into the aorta, and that's what the ductus arteriosus does. This becomes the ligamentum arteriosum in the newborn heart. And so the ligamentum arteriosum, it's kind of like a little bungee cord, to be honest with you. It's what straps together the pulmonary arteries and the aorta. There's just this little white chunk of tissue there that sort of just holds everything together because it's like a garden hose. If you've ever turned a garden hose on, at first it kind of recoils and bounces around a little bit. It's the same kind of thing. Those vessels, as that blood is being pushed through there every single heartbeat, it can cause all those vessels to move around too much. So it just sort of holds everything together. The ductus venosus, this is what shunts oxygenated blood from the umbilical vein to the inferior vena cava. After birth, it becomes the ligamentum venosum. So the ductus venosum is down at the navel, right? And so you can feel the remnants of your ductus venosus if you stick your finger in your navel and press up. You'll feel a cord right there, and that has become the ligamentum venosum. Sorry about the typos in there. These adaptations maintain oxygen and nutrient delivery via the placenta and allows you to circumvent the inactive fetal lungs. So there's what it looks like in a fetal heart. So in a fetal heart, the blood is coming in all the way down there at the bottom, and the blood is coming in oxygenated because mom oxygenates the blood for the baby, and it's going to run through the liver, and in the liver, it's going to drop off nutrients, and we'll talk about why that's important later because that's where your blood is going to make sure that nutrients get out to all your cells. But nonetheless, it's going to drop off some nutrients there and feed the baby. And then it's going to head up the inferior vena cava, which in this case is red because it's oxygenated, and it's going to shoot right through that foramen ovale and head over to the aorta and out to the rest of the body. So you see that we somewhat circumvent that whole pulmonary route. Now notice it's pink, which means that there is a little bit of blood that goes out. We do have developing cells out in the lungs, and they do need oxygen, but the in the adult, the point of pulmonary circulation is to reoxygenate the blood, and in the fetus, we aren't doing that. So there's a small amount that goes up. They also show you that the other shunt, the ductus arteriosus, if too much blood is headed out there to the lungs, there's some of it that just gets bypassed back into the aorta and sent on its way. And then you see the shunt down at the bottom in the liver, the ductus venosus. All right. So that is the information for this particular chapter. You want to make sure you study all this. You want to make sure things that you don't remember from 210, you go back and look at those in the lectures that are available to you. You want to make sure that you do your Connect assignments, which are more tied into lab. But this week, the lab is on hearts and vessels, and so are the lectures. So those two kind of tie together. You have quizzes to take, and I will see you all for the vessel lecture. Bye.